this video, we're going to do a tour of the cell. So by the end of this lecture, you will be able to label all of these structures and understand their functions. Let's get started. First of all, what is a cell? Because all living things or organisms are built from cells. Cells are the basic structural units of living organisms, and there are two different cell types, prokaryotic and eukaryotic cells. Prokaryotes are divided into two domains, which are bacteria and archaea, and these cells do not have a nucleus. And eukaryotes include plants, animals, protists, and fungi, and these cells have a nucleus. Eukaryotic means true nucleus. Eukaryotic cells are much larger than prokaryotic cells. We're going to be mainly focusing on eukaryotic cells, but we'll touch on some features involving prokaryotes as well. Eukaryotes share one common feature, and that is they all contain membrane-bound organelles that form these specialized compartments within the cell. This is known as cell compartmentalisation, and the benefit of this is that it allows processes and reactions that require different environments to occur at the same time in the same cell and at the right conditions. The membrane-bound compartments within eukaryotes are called organelles, and the membranes around the organelles control the movement of substances between the organelle and the cell cytosol. Let's start with the cell membrane before touring the structures inside. Okay, what I've done here is on the top right hand corner, you'll be able to see what structure we're talking about. And later on, all the other parts will be colored in gray except the part we're talking about. All cells are bounded by a selective barrier called the plasma membrane or the cell membrane. Okay, but look at this masterpiece for a second. All cells contain cytosol, a jelly-like material in which subcellular components are found. It's the intracellular fluid. And the region between the nucleus and the plasma membrane is called the cytoplasm. It's the entire contents within the cell membrane, including the organelles. Let's zoom in on the plasma membrane, which functions as a selective barrier that allows passage of nutrients and waste. The plasma membrane and the membranes of organelles consist of a double layer or bilayer of phospholipids with various proteins embedded in it. We have some globular proteins, peripheral, integral, and glycoproteins shown here, and a protein channel. Let's zoom in on the structure of a phospholipid. The structure of a phospholipid consists of a polar head or hydrophilic head, water-loving, and two hydrophobic or non-polar hydrocarbon tails. It's scared of water. Therefore, when phospholipids are added to water, they form a double-layered sheet called a bilayer that protects their hydrophobic tails from water because it's scared of water. The hydrophobic parts are found in the interior of the membrane, okay, this right here, while the hydrophilic regions are in constant contact with aqueous solutions on either side of the cell. So the bilayer forms a wall between the cell and its external environment. Now, it's also important to note that the membranes participate in the cell's metabolism because many enzymes are built into the membranes. So that's the cell membrane. Inside the cell, we can find different organelles, and each of these organelles have a very specific structure and function. So now let's subtract complexity and break down the two components involved in the genetic control of the cell, the nucleus and the ribosomes. The nucleus contains most of the cell's DNA, or genetic material. Prokaryotes do not have a nucleus, which means there's no barrier that separates the DNA from the cytoplasm. So then where is it stored? Let's go through this. The genetic material of prokaryotes is called the genophore, which is a single circular DNA chromosome contained in a region known as the nucleoid. So the nucleoid doesn't have a nuclear membrane, and many prokaryotes also contain plasmids, okay, which are small rings of double-stranded DNA that carry a few genes. Now switching to eukaryotic cells, eukaryotes have a nucleus that contains most of the genetic material. You can also see the endoplasmic reticulum here, studied with ribosomes, because the structure of the nucleus is continuous with the ER. Here's the nuclear envelope. It's a double membrane that separates the content of the nucleus from the cytoplasm. The two membranes are a lipid bilayer, and the nuclear envelope has pore structures that are lined by a protein structure called a pore complex that is important in regulating the entry and exit of proteins, RNAs, and macromolecules. And then we have the nuclear lamina, a net-like network of protein filaments, intermediate filaments, or protein meshwork that surrounds the nucleus. So it's going to line the inner surface of the nuclear membrane. This structure helps maintain the shape of the nucleus by mechanically supporting the nuclear envelope. Let's zoom in to what's within the nucleus. DNA carries the genetic information of the cell. It consists of two long polynucleotide chains. Recall what a polymer is. It's a molecule consisting of many similar subunits that are linked together by covalent bonds, in which one or more pair of valence electrons are shared. Each chain is composed of 4 types of nucleotide subunits, and the 2 strands are held together by hydrogen bonds between the base portions of the nucleotides. And if we zoom in to the structure of nucleotides, it's composed of a nitrogen-containing base or nitrogenous base, adenine, guanine, cytosine, or thymine. The sugar in DNA is deoxyribose, and a phosphate group is attached. Now, in eukaryotic cells, DNA molecules are packaged into chromosomes. Each chromosome consists of a single long linear DNA molecule associated with proteins that fold and pack the DNA into a more compact structure. The structure of DNA and tightly bound protein is known as chromatin. Okay, so we have condensed chromatin here. So chromosomes organize and carry genetic information. Specifically, they carry genes, which is the basic unit of inheritance. It's the segment of DNA that contains instructions for making a particular protein, which we'll touch on in a second. Now, the total genetic information present in an organism constitutes its genome, all right? Referring to human cells, each human cell nucleus contains 2 copies of each chromosome, one inherited from the mother and one from the father. Now, this doesn't apply to gametes, sperm and eggs, and highly specialized cells that lack DNA entirely, such as mature red blood cells. Each human cell contains a total of 46 chromosomes, including 22 pairs that are common in both males and females, called autosomes, and 2 sex chromosomes, X and Y in males and 2 X's in females. So that's what's inside the nucleus. We cover this in great detail in the DNA structure and function lecture. Now, another important structure in the nucleus is the nucleolus, which plays an important role in the transcription and processing of ribosomal RNA or rRNAs. Proteins from the cytoplasm are assembled here with ribosomal RNA into large and small subunits of ribosomes. These subunits then exit the nucleus through the nuclear pores into the cytoplasm, where they can be assembled into a ribosome. You'll see this connection in the translation lecture. It's absolutely gorgeous when you see the overview picture of how it's carried out. Now, we've covered the structure of the nucleus and what we can find within it, but what else does it do? The nucleus also directs protein synthesis. Let's break this down. We start with DNA in the nucleus, and this right here is the nucleotide sequence of a gene, okay? It's a segment of DNA that contains instructions for making a particular protein. When a particular protein is needed by the cell, this segment of DNA is first copied into RNA, specifically messenger RNA or mRNA. The mRNA is then transported to the cytoplasm via nuclear pores, and once it's in the cytoplasm, the RNA copies are then used to direct protein synthesis. Ribosomes translate the mRNA's genetic message into a polypeptide. So the flow of genetic information in cells is from DNA to RNA to protein. And this process is called gene expression. The information encoded in the DNA sequence is converted into either a protein or just RNA. And that leads us to the next structure, which are ribosomes, or the protein factories of the cell. Like we mentioned before, ribosomes carry out protein synthesis. A ribosome consists of a large subunit and a small subunit, each made up of proteins and one or more ribosomal RNAs or rRNAs. In both bacteria and eukaryotes, a large and small subunit join to form a functional ribosome only when attached to an mRNA molecule. Now, ribosomes are not considered organelles because they are not membrane bound. They don't have a membrane. There are two regions where ribosomes are found in the cytosol. There's free ribosomes in the cytosol, or they're attached to the outside of the endoplasmic reticulum or nuclear envelope, and these ribosomes are called bound ribosomes, okay? So both types are structurally identical. Cells with high rates of protein synthesis have large numbers of ribosomes. Let's take a closer look at the structure of ribosomes and see how they operate in the process of translation or protein synthesis. So we have the large ribosomal subunit and the small subunit, and then here's the mRNA binding site. The ribosome contains 3 binding sites for tRNA molecules called the A site, the P site, and the E site. The A site is the amino-acyl tRNA binding site, which holds the tRNA carrying the next amino acid. Okay, so a charged tRNA enters the A site by base pairing with the complementary codon on the mRNA molecule. We covered this in the transcription and translation lecture. The P site is the peptidyl tRNA binding site, which holds the tRNA carrying the growing polypeptide chain, and the E site is the exit. So the discharged tRNAs leave the ribosome from the E site. This cycle of reactions is repeated each time an amino acid is added to a polypeptide chain. Absolutely gorgeous. Okay, let's quickly summarize the 2 components before continuing with our tour. The genetic instructions are located in the nucleus, and protein synthesis is carried out by the ribosomes. All right, let's continue with our tour. Now, many of the different membrane-bounded organelles are part of the endomembrane system, which includes the plasma membrane, nuclear envelope, the endoplasmic reticulum, the Golgi Apparatus, lysosomes, and numerous types of vesicles and vacuoles. The endomembrane system performs a multitude of functions in the cell, including protein synthesis, protein transport into membranes and organelles or out of the cell, and lipid metabolism. The membranes are linked either by direct physical continuity or through membrane segment transfer as small vesicles, sacs made of membrane. Now, we're going to break down what's going on here because there's quite a lot and what these arrows are doing and how this all works. But let's go through each structure and their functions. Starting with the endoplasmic reticulum, which is a vast network of membranes that make up more than half of the total membrane. The word endoplasmic means within the cytoplasm. So the ER is made up of cisternae, which are a network of membrane tubules and sacs. The ER membrane divides the cytosol from the ER lumen or cavity, which is the interior compartment of the ER. The space between the 2 envelope membranes is continuous with the ER lumen, and the ER membrane is continuous with the nuclear envelope. Now, there are 2 regions of the ER called the smooth ER and the rough ER, and have different structures and functions. The smooth ER lacks ribosomes on its outer surface, hence why it's called the smooth ER, because it's smooth, whereas the rough ER is studded with ribosomes, giving it a rough appearance under the electron microscope. As we mentioned earlier, ribosomes are also connected to the cytoplasmic side of the nuclear envelope's outer membrane, which is continuous with rough ER. Let's go through their functions. The smooth ER is involved in a variety of metabolic processes that differ depending on the cell type. These include lipid synthesis, carbohydrate metabolism, and drug and toxin detoxification. Smooth ER enzymes play a key role in the synthesis of lipids, such as oils, steroids, and new membrane phospholipids. Sex hormones in vertebrates and different steroid hormones secreted by the adrenal glands are some of the steroids produced by the smooth ER in animal cells. Okay, another function is that it stores calcium ions. The smooth ER membrane transports calcium ions from the cytosol into the ER lumen in muscle cells. So calcium ions flow back over the ER membrane into the cytosol when a muscle cell is stimulated by a nerve impulse, causing the muscle cell to contract. The release of calcium ions from the smooth ER causes a variety of responses in other cell types, including the secretion of vesicles containing newly generated proteins. All right, that's the smooth ER. Let's move on to the functions of rough ER. Ribosomes connected to rough ER generate proteins, which are secreted by many cells. For example, specific pancreatic cells produce the protein insulin and secrete it into the bloodstream. So as the polypeptide chain grows from a ribosome, a bound ribosome, the polypeptide chain is threaded into the ER lumen through a channel created by a protein complex in the ER membrane. And as the new polypeptide enters the ER lumen, it folds into its functional shape. Now, glycoproteins, which are proteins that have carbohydrates covalently attached to them, make up the majority of secretory proteins. Enzymes embedded into the ER membrane connect carbohydrates to proteins in the ER lumen. Now, after secretory proteins are created, the ER membrane separates them from proteins synthesized in the cytoplasm by unbound ribosomes or free ribosomes. The secretory proteins leave the ER wrapped in the membranes of vesicles that emerge from a particular region known as the transitional ER. They look like bubbles, okay? Vesicles that transport from one section of the cell to another are called transport vesicles, which we'll discuss in a second. Another function of rough ER is it serves as a membrane factory for the cell, growing in place by adding membrane proteins and phospholipids to its own membrane. And polypeptides destined to be membrane proteins are introduced into the ER membrane and held there by their hydrophobic regions as they develop from ribosomes. And just like the smooth ER, the rough ER also produces membrane phospholipids. Enzymes embedded in the ER membrane assemble phospholipids from cytosol precursors. And so the ER membrane grows and sections of it are transported to other components of the endomembrane system in the form of transport vesicles. Okay, so we toured the structure and function of the endoplasmic reticulum. Let's now move on to the shipping and receiving center of the cell called the Golgi Apparatus. After leaving the ER, many transport vesicles travel to the Golgi Apparatus. The Golgi can be viewed as a warehouse that handles receiving, sorting, shipping, and even manufacturing. Isn't it incredible how the cell is constructed? It's breathtaking, honestly. So, proteins and other ER products are altered and stored here before being transported to other locations. The Golgi apparatus is unusually vast in cells that specialize in secretion. The Golgi apparatus is made up of a stack of flattened membrane sacs called cisternae. Each cisternae in a stack has a membrane that divides its interior space from the cytoplasm. Now, a Golgi stack receives and dispatches transport vesicles and the products they contain. It has structural and functional directionality, with a cis phase that receives vesicles containing ER products and a trans phase that dispatches vesicles. The term cis means on the same side, and this side is usually located near the endoplasmic reticulum. Transport vesicles move material from the ER to the Golgi apparatus, so a vesicle from the ER can add its membrane and the contents of its lumen to the cis phase by fusing with the Golgi membrane on that side. Now, trans phase means on the opposite side. So the vesicles coming from here travel to other sites. And ER products are usually altered during their transit from the cis to the trans region of the Golgi apparatus. So the Golgi produces and refines its products in phases, with various cisternae carrying distinct enzymes. The name for this is the cisternal maturation model, in which the cisternae of the Golgi actually progress forward from cis to the trans phase, carrying and altering their content as they move. So before a Golgi stack sends its products out by budding vesicles from the trans phase, it's going to sort them out and direct them to different regions of the cell. They can add molecular identification tags, such as phosphate groups, and these transport vesicles may also have external molecules on their membranes that recognize docking sites on the surface of certain organelles or on the plasma membrane, allowing them to be properly targeted. Amazing. That's the Golgi apparatus. The next organelle we're going to look at are lysosomes. Many eukaryotic cells use lysosomes to hydrolyze or break down macromolecules. What exactly is a lysosome though? A lysosome is a membrane sac containing hydrolytic enzymes. The enzymes, called lysosomal enzymes, work best in the acidic environment found in lysosomes. So let's say a lysosome breaks open or loses its content. The released enzymes are not highly active because the cytosol has a near-neutral pH. Excessive leaking from a high number of lysosomes, on the other hand, can cause the cell to actually self-destruct. All right? How are these enzymes and lysosomal membranes made? They are made by rough ER and then transferred to the Golgi apparatus to be processed further. In many different situations, lysosomes perform intracellular digestion. Let's go through this. Amoebas and many other unicellular protists eat by absorbing smaller organisms or food particles, a process called phagocytosis. A lysosome fuses with a food vacuole here during the process of phagocytosis by a unicellular protist. The lysosome contains enzymes that break down the food. The subunits, such as simple sugars and amino acids, enter the cytosol and provide nutrients for the cell. So here we have the digested food. Phagocytosis is also carried out by some human cells. For example, macrophages are a type of white blood cells that helps the body protect itself by engulfing and eliminating bacteria and other invaders. Okay, that's phagocytosis. There's also autophagy, which is another type of process that lysosomes carry out, in which they use their hydrolytic enzymes to recycle the cell's own organic material. That's lysosomes. The next part we're going to look at are vacuoles. What are vacuoles? Vacuoles are large vesicles produced by the endoplasmic reticulum and the Golgi apparatus. Vacuoles provide a variety of functions in many cell types. For example, the formation of food vacuoles via phagocytosis, which we've already covered. In plants, small vacuoles can store essential chemical components such as proteins stored in storage cells in seeds. They may also help protect the plant from herbivores by storing harmful substances for animals. Mature plant cells typically have a big central vacuole formed by the convergence of smaller vacuoles. The solution inside the central vacuole, called the cell sap, is the plant cell's primary reservoir of inorganic ions such as potassium and chloride. Okay, but what is the importance of this central vacuole? This is crucial in plant cell growth because it collects water, allowing the cell to expand larger with little investment in new cytoplasm. So we've covered the endomembrane system. Let's take everything together and review the flow of membrane and proteins through the various organelles. All right. So we have the nucleus envelope here, which is connected to the rough ER. It's continuous with both the rough and smooth ER. The membranes and proteins that are produced by the ER move to the Golgi via transport vesicles, okay? And here, the Golgi apparatus produces transport vesicles and other vesicles that produce lysosomes and vacuoles. The lysosome here confused with another vesicle for digestion. The cis Golgi is the receiving side and the trans Golgi is the shipping side, and a transport vesicle from the Golgi, which contains products, is transported to the plasma membrane for secretion, and the products are secreted by exocytosis. All right? And we also have the summary table here, showing the structure and function of each structure. Moving forward with the next part of the tour, let's take a look at the organelles that convert energy to forms that cells can use for work: mitochondria and chloroplasts. Mitochondria are the sites of cellular respiration, which is the metabolic activity that uses oxygen to generate ATP by extracting energy from carbohydrates, lipids, and other fuels, the powerhouse of the cell. On the other hand, chloroplasts found in plants and algae are the sites of photosynthesis. So by absorbing sunlight and using it to drive synthesis of organic substances such as sugars from carbon dioxide and water, chloroplasts transform solar energy into chemical energy. Okay, let's break down the structure and function of mitochondria first. Mitochondria can be found in almost every eukaryotic cell, including those of plants, animals, fungi, and most protists. Some cells have a single big mitochondrion, but most have hundreds or thousands. This correlates with the metabolic activity of the cell. Cells that move or contract, for example, have proportionally more mitochondria per volume than cells that are less active, because they need more power, they need more energy. Each of the two membranes is a phospholipid bilayer with a different set of proteins embedded in it. The outer membrane is smooth, while the inner membrane is convoluted, containing infoldings known as cristae. The inner membrane divides the mitochondrion into two internal compartments. First is the intermembrane space, which is the thin space between the inner and outer membranes. And the inner membrane encloses the second compartment, the mitochondrial matrix. Many different enzymes, as well as mitochondrial DNA and ribosomes, are found in the matrix. Some of the processes of cellular respiration are catalyzed by enzymes in the matrix. And other proteins involved in respiration, such as the ATP-producing enzyme, are embedded in the inner membrane. Okay, let's now take a look at chloroplasts. Okay, we'll look at this masterpiece first. Chloroplasts include the green pigment chlorophyll, as well as enzymes and other components involved in the photosynthetic synthesis of sugar. A chloroplast's contents are separated from the cytoplasm by an envelope made up of two membranes separated by a very small intermembrane gap. And another membrane system exists inside the chloroplast in the shape of flattened link sacs known as thylakoids. Thylakoids are stacked like poker chips, or I like to see them as cookies, okay? Each stack is termed a granum. And the stroma is the fluid that surrounds the thylakoids and includes chloroplast DNA, ribosomes, and a variety of enzymes. And then we have the lumen. So the membranes of the chloroplasts divide the chloroplast space into three compartments. We have the intermembrane space, the stroma, and the thylakoid space. And this organization allows the chloroplast to convert light energy to chemical energy during photosynthesis. So here's a quick summary of the structure and function of mitochondria and chloroplasts: cellular respiration and photosynthesis. Okay, let's move on to the next organelle, peroxisomes. The peroxisome is a highly specialized metabolic compartment with a single membrane. Peroxisomes include enzymes that remove hydrogen atoms from a variety of substrates and transfer them to oxygen, producing hydrogen peroxide as a byproduct. This is where the organelle gets its name, because it produces hydrogen peroxide. These reactions serve a variety of purposes. Some peroxisomes use oxygen to break down fatty acids into smaller molecules, which are then transferred to mitochondria and used as cellular respiration fuel. Another example includes alcohol, and other toxic substances are detoxified by peroxisomes in the liver, which convert hydrogen from the dangerous molecule to oxygen. Now, peroxisomes produce hydrogen peroxide, which is harmful. But the organelle also includes an enzymes that transforms hydrogen peroxide to water, okay? The enzymes that create hydrogen peroxide and those that dispose of these toxic substances are isolated away from other cellular components that could be damaged. So this illustrates how the compartmental organization of the cell is very important to its function, okay? So we've toured the genetic storehouse, the endomembrane system, and the powerhouse of the cell. Let's now go through the network of fibers that organizes structures and activities in the cell, the cytoskeleton. The cytoskeleton provides mechanical support and structure to the cell. This is very important for animals because they lack walls, so the cytoskeleton serves as an anchor for many organelles. It's involved in motility and signal transmission. Let's look at the three primary types of fibers that make up the cytoskeleton. We have microtubules, which are the thickest of the three types, microfilaments, also known as actin filaments, and they are the thinnest, and then we have the intermediate filaments. Starting with microtubules, microtubules are hollow rods made up of globular proteins called tubulins that are found in all eukaryotic cells. They form and support the cell as well as serve as tracks for organelles with motor proteins to travel along, okay? So here we have motor proteins that travel along the microtubule. Microtubules develop from a centrosome, which is commonly found around the nucleus in animal cells. Within the centrosome is a pair of centrioles, okay? These right here. Each centriole is made up of 9 pairs of triplet microtubules organized into a ring. Now, Although centrosomes within centrioles may assist in the organization of microtubule assembly in animal cells, many other eukaryotic cells lack centrosomes with centrioles, and so they organize microtubules in different ways. We can also find microtubules in some eukaryotic cells that have flagella and cilia. The beating of these structures is caused by a particular arrangement of microtubules. Okay? That's microtubules. The next type of fiber are microfilaments or actin filaments. They are present in all eukaryotic cells. These are thin solid rods, and the reason why they are called actin filaments is because they are made from molecules of actin, which is a globular protein. Microfilaments function in muscle contraction. Thousands of actin filaments and thicker filaments made up of a protein called myosin interact. Okay, so this right here. Okay, so that's actin filaments. The third type are intermediate filaments. Why are they called intermediate filaments? Because their diameter is larger than the diameter of microfilaments, but smaller than that of microtubules. It's just in the middle. It's the middle child. Now, while all eukaryotic cells include microtubules and microfilaments, intermediate filaments are only found in the cells of a few animals, including vertebrates. Each kind is made up of a specific molecular subunit that belongs to a protein family that includes the keratins. Intermediate filaments support cell shape and fix organelles in place. They are often disassembled and reassembled in various regions of a cell. So intermediate filament networks typically exist after a cell dies. For example, the outer layer of human skin is made up of dead skin cells filled with keratin filaments. Alright, that's the cytoskeleton. Here's the table summary for peroxisomes and the cytoskeleton. Now, before we end this lecture, let's take all the structures we've covered and label these animal and plant cells. I'm sure you'll be able to label them and know their function as well. We have the nucleus, which contains genetic heredity information, the ribosome, which synthesizes proteins, the rough and smooth ER, which processes and modifies proteins, and for the smooth ER, it synthesizes lipids, the Golgi Apparatus. Okay, this is the warehouse of the cell. It processes and packages proteins. Lysosome, which digests cellular waste material. We have the vacuoles, which store substances. The powerhouse of the cell, mitochondria, which obtains energy from organic compounds, from the oxidation of food molecules such as sugars to produce ATP. Then we have chloroplasts in plant cells, which uses light energy, carbon dioxide, and water to produce glucose, okay, photosynthesis. And then we have peroxisomes, which contains enzymes involved in several metabolic reactions. And then the cytoskeleton, which helps cells maintain their shape and internal organization. And then we also have the centrosome. Thank you for watching this video. Make sure you subscribe to EKG Science so you don't miss a single lecture. And remember, subtract complexity and slow down. To study the next lecture, simply click the next video or you can view the entire playlist. Hey, stop procrastinating.